Hello, welcome back to Regulatory Affairs 101. I am Nancy. Today, we're diving into one of the most important areas in modern biologics development, biosimilar products. We'll walk through the regulatory foundations, development pathways, global frameworks, and the latest FDA considerations for container closure systems and device constituent parts. Let's get started. A biosimilar is a biological product that is highly similar to an FDA-approved reference product, with no clinical meaningful differences in safety, purity, or potency. Minor differences in inactive components can exist, but the clinical performance must match. In the United States, biosimilars are approved under Section 351K of the Public Health Service Act, the abbreviated pathway designed specifically for biosimilar and interchangeable products. Biosimilars and generics both expand access, but they are fundamentally different. Biosimilars are complex biologics made in living systems, while generics are small molecules made through chemical synthesis. The regulatory pathways differ: 351K for biosimilars and 505J for generics. Biosimilars require analytical, non-clinical, and clinical data, whereas generics rely mainly on bioequivalence. And importantly, biosimilars need a specific FDA interchangeability designation for substitution, while generics are generally substitutable under state law. There are four core concepts in biosimilar regulation. First, high similarity demonstrated through extensive analytical characterization. Second, the totality of evidence, meaning FDA evaluates all data together. Third, extrapolation, which allows approval for additional indications when scientifically justified. And fourth, interchangeability, which requires data showing safe and effective switching. The FDA uses a stepwise approach. We begin with analytical studies, structural and functional characterization, identification of critical quality attributes, and evaluation of the complete product presentation, including container closure and device components. Next are non-clinical studies, such as mechanisms of action and in vitro functional assays. Finally, clinical studies focus on PK/PD similarity, immunogenicity, and, when needed, a comparative clinical trial. Analytics always lead the way. Interchangeability allows pharmacy substitution of the reference product subject to state law. To earn this designation, sponsors must conduct switching studies demonstrating no increased safety risk and no reduction in efficacy. Interchangeability is not required for biosimilar approval. And for products delivered via devices, FDA expects sponsors to evaluate whether device design or user interface differences could affect safe switching. Biologics are highly sensitive to manufacturing conditions, cell line, culture environment, and purification processes all matter. Manufacturers must demonstrate strong process control, consistency, and product comparability. Post-approval changes follow established CMC lifecycle management principles. And container closure systems and device constituent parts must be evaluated as part of overall product quality and usability. FDA's 2026 draft guidance adds important clarity for biosimilars that use container closure systems or device constituent parts. Product presentation must match the reference product in dosage form, strength, route of administration, and conditions of use. Sponsors must provide complete CMC information for container closure systems and device components, including extractables and leachables, performance testing, and stability data. For combination products like prefilled syringes or auto-injectors, FDA expects comparative analysis of the user interface, including physical comparison, task analysis, and label comparison. Analytical characterization is the foundation of biosimilar development. This includes primary structure, higher order structure, glycosylation profile, biological activity, impurity profile, and stability and degradation pathways. These data form the backbone of the similarity assessment. In clinical development, PK/PD similarity is often the primary requirement. Immunogenicity assessment is essential. Comparative efficacy trials may be reduced or waived when analytical similarity is robust. Sponsors should select sensitivity populations and endpoints to detect potential differences. FDA may extrapolate indications when several conditions are met. The mechanisms of action is supported across indications. Receptor binding and PK/PD are comparable. Immunogenicity and safety concerns are addressed. And the totality of evidence supports use in the extrapolated population. Globally, biosimilar frameworks share common principles but differ in implementation. The EMA launched the first biosimilar pathway in 2005 and maintains strong analytical requirements. Health Canada aligns closely with EMA and permits extrapolation. PMDA often requires local clinical data, and World Health Organization provides harmonization principles for countries establishing biosimilar pathways. In the US, non-proprietary names include a four-letter suffix. For example, infliximab DYYB to support traceability. Labeling includes a biosimilarity statement, and interchangeability claims appear only when designated. The clinical data of reference product may be included. Post-marketing safety monitoring is essential for biologics. Distinct suffixes support traceability. Immunogenicity surveillance continues throughout the product lifecycle. Risk management plans, or RIMs, may apply depending on the product. Pricing and reimbursement vary by region. Payer formulary decisions strongly influence uptake. Education is critical to building prescriber and pharmacist confidence. Real-world evidence continues to support biosimilar adoption. Examples of successful biosimilar adoption include infliximab biosimilars such as Inflectra and Renflexis. Multiple adalimumab biosimilars launched in 2023 and 2024, and trastuzumab biosimilars in oncology. These products have demonstrated comparable outcomes and meaningful cost savings. Challenges include manufacturing complexity, provider hesitancy, and confusion around interchangeability. Opportunities include lower healthcare costs, expanded patient access, and advances in analytical technologies. In summary, biosimilars expand access to biologic therapies. Approval is based on the totality of evidence, with analytical similarity as the foundation. Interchangeability remains a distinct destination, and global regulatory alignment continues to evolve. Before we wrap up, here's a quick knowledge check. Which statement best describes the foundation of biosimilar approval under the FDA's 351(k) pathway? A. Biosimilars are approved primarily based on large comparative clinical trials. B. Biosimilars must demonstrate identical manufacturing processes to the reference product. C. Biosimilars are approved based on the totality of evidence with analytical similarity as the foundation. D. Biosimilars are automatically interchangeable once approved. The correct answer is C. Biosimilars are approved based on the totality of evidence with analytical similarity as the foundation. These references include FDA, EMA, Health Canada, PMDA, and World Health Organization guidance documents, along with educational resources such as the Biosimilars Forum and FDA's Biosimilars curriculum. They are excellent sources for deeper study. Thank you for watching. If you like the content of this video, please like, subscribe, and comment below. Have a wonderful day.